And thank you everybody for coming, and congratulations all the human scholars and the human mentors. You had a wonderful accomplishment and journey for this summer program, and thank you for your devotion and all the effort by joining each labs and our roundtables. And thank for the mentors and all the professors and postdocs who have provided lectures on the roundtable. So, and we also want to thank the INDR Foundation providing support, and our chair, Dr. Sahal, and all the facilities supported by the Department of Ophthalmology, and also the Human Academy, including David Boone and Solomon and Stephen. There's a lot of support here, and we seems after COVID we have really recovered from partially Zoom last year and completely in person this year. I would like to start my presentation about the closing ceremony. That I will do it again in the afternoon, but because some of the people won't be here in the afternoon. So I don't know why this is showing. I need to swap. Okay, good. Okay, so this is our ophthalmology site. This is our second year joining the human program. And we have several mentors continue 2 years as a mentor, and really appreciate, including Dr. Genji and Dr. Gary and Dr. Mayo and yeah, everybody basically. And I have a slide here. So that's a small reminder of first lecture about the amazing structure of the eyes. So this is what everybody's doing. What's going on? I'm sorry for my mistake, but this is Dr. Shaohua Pi, and there's it's awkward. We want to thank everybody here, including Dr. Michelle Genji, the physician, and Dr. Gary Yam, research scientist, and Dr. Patrick Mayo, Dr. Shaohua Pi, I will correct this, and myself, and Dr. Robert Shanks and Dr. Du all participated as human mentors who have engaged not only themselves, but also some of their lab members on everyday activity for each of you. And we also want to thank all the trainees and scholars here who have participated, really get your feet wet and put your hands on some real experiments, including some very cool imaging analysis, molecular biology, some animal shadowing and handling, and another example of imaging analysis, and very cool video games activity, and studies of how you engage your vision and your eye movement during playing a very cool video game. And also very cool engineer projects on 3D printing. I guess you will see very nice gadgets and toys later on. So these are a brief summary of what's going on for our roundtable seminars. We have started a really nice program this semester that we included some scientific lectures, including introduction of the eye, how our brain works to process vision, and what's happening during different retinal diseases, and how each typical retinal diseases look like. And we have great scientists talking about scientific career, and also we have a great discussion with Ear and Eye Foundation people. And we have 2 lectures talking about how to make a scientific presentation and how you utilize online tools to search for relative scientific information and literature. And so these are the very cool research projects each of you have been participating, including eye movement tracking, and molecular biology and imaging and engineering. So really covering a diversity of the real research programs that's ongoing within the department. And while we are doing science, we also enjoy and tasting different type of food. And yeah, I think hopefully you enjoyed it. And there are people on the back of the scene that's provide a lot of support, including the department's admin and also our chair who is really supportive to this foundation and to this research program. He will show up later on because there's a clash of another meeting happening at the convention center. And also, I think all of you have, the students have met the Eye and Ear Foundation people who is really working very hard to look for supports for the department and for this program. And also, we want to thank other people that participate directly or indirectly, including the lab members in each mentor's lab, and our admin who has been working hard on figuring out the accounting for this program, and medical doctors giving us lectures, and research professors giving lectures, and postdocs sharing their life. And we had a picnic last week and really had some fun. So that's it for me, and wish you had all very nice summer. And I would like to ask if any mentor would like to say something and add on to. Yeah, Heather.
Yes. Some of you guys
Heather, please come on.
Okay,
And
yes. I never met a podium I didn't like. Okay. So I'm Heather Kronos-Danek with the Eye and Ear Foundation. Some of you, I got to see Skyler and Samuel this morning, and I get to see so many people I work with. Everyone else at the foundation, including Dr. Sahal, are at the Mission of Mercy event at the Convention Center, which is another part of our very important work. Not only do we fund research, we support the clinical and community initiatives that Dr. Sahal sets forth. Over 1,000 patients are going to be seen over the next 2 days who need dental care, eye care, and hearing tests free of charge. And it's a very vital thing that we do. But I want to thank the Hillman Family Foundation for this program. None of this would have happened without Hillman. And none of this would happen unless we had the amazing, amazing chair, Jose Sahel, who's put together a really dynamic research and clinical team. These mentors that you all have, they're so excited for you. And I'm excited for you, like the energy. You know, Skyler was up here and she's like, yeah. When I saw her presenting this morning, that's good stuff. So thank you to all of you. I've got to take off to go help at the Mission of Mercy event, but all of you have a big future ahead of you, a long road in your career, and good luck to each and every one of you. And it's been a great experience for us as a foundation to watch you. Thank you.
Now, anybody else? Any students who want to say something before your formal presentation? Share with us.
We'd like to thank Dr. Chen for all of this organization.
Thank you for all the I really want to appreciate all the mentors who voluntarily get involved in this program. I know it's really also learning experience for everybody of involving. Many of us have been for the first time mentoring high school students who are a little bit different from our previous trainees that are commonly undergrads or postdocs or PhD students. So really appreciate your patience and you're willing to reach out and help for the young talents in the future. So
I'm just going to share, I'm a Glen teacher, Chen, I'm a teacher at Excitebike, and two of the trainees, and I don't know if any other teachers are here, but I just would like to say thank you to all of you as colleagues, as mentors, as students for making this happen, because if you don't really understand the impact, I see it in the classroom, it's huge, and to see the students get up there and speak the language after their experience, it's kind of brings tears to us. So thank you very much for your time, because I know it's
for coming. And let me start with the, so who is the first one? Please step, Skyler, right? Who is the Skyler's mentor? Yeah, Gary, please. Gary, how do I, please let your mentor introduce you.
Oh, okay. So yeah, okay. So good morning, everyone. I'm Gary M. So I'm PI in the corneal degeneration lab. So I'm the mentor of Skyler Spears. So it's a really great pleasure to have her in my lab for the last 6 to 7 weeks, which is a very good time. And because she's a very nice girl, she works hard and she knows what she's doing and what she needs to do. So that's why we, with the help of Maura, because she also contributed a lot in Skyler's project of doing the 3D reconstruction from the OCT images and also the measurement and so on. So fortunately that we can get something that is presentable. Yeah, even though the time is very short, but she works very hard and she's very talented and she really tried to do something in this short period of time. So, so I think right now, let her speak to you, what she has been done, she has been doing, and also what she obtained and what her experience in our lab, how she.
This is the screen. So.
Okay, okay. Yeah. So her talk will be the application of an OCT to based 3D reconstruction model. For quantitative assessment of corneal changes after the stem cell therapy, which we have been doing in our lab for a kind of alternative treatment for corneal scarring. Yeah. So Skyler, let's check the floor. Yeah.
Hello.
Oh, more. Come here. Come. You are the most important mentor.
Probably mask off for one.
Hello, my name is Skyler, and my presentation is the application of an OCT-based reconstruction model to a quantitative assessment of corneal changes after stem cell therapy. I attend Pittsburgh Science and Technology Academy, and my mentors in the lab were Dr. Gary Yam and Ms. Maura Gary. My mentor in the clinic was Dr. Vishal Janji, and I was a part of the corneal regeneration lab. So let me introduce you to the cornea. There are 5 different layers of the cornea, which are the epithelium. Okay, let me do this again. So there are 5 different layers of the cornea, but there are 3 different cell layers, which are the epithelium, the stroma, and the endothelium. There are also 2 different membranes included in the cornea. So that will be the basement membrane and the descendants membrane. So the epithelium is on the outermost layer of the cornea and it's used for protection. The stroma is in the middle of the cornea and it is the thickest layer and it's used for strength, transparency, and refractivity. The endothelium is on the innermost layer of the cornea and is used to regulate hydration. So when there's damage in the stroma, that can cause haze or blindness in the eye. And the current treatment for that is to get a corneal transplant. The issue with corneal transplants is that we don't have enough donors for the amount of patients that need it. So my lab created an alternative to transplants, which is cell therapy using stem cells treatment. So basically what we do is we take the donor cornea and we expand their cells so that we can use it for multiple recipients, which is shown here. This is what happens when we do the cell therapy, and this is how it would be if they just did the stem cell, if they just did the regular treatment. So corneal anterior stromal injury caused by mechanical ablation using algebra burn. So this is the algebra right here, and we use Balsi mice and we put them under anesthesia. So the first thing we do with the algebra ablation is we remove the epithelium, which is shown right here, and then we do a second ablation, which is to injure the anterior stromal layer. So after doing so, we apply stem cell suspension and fibrin gel. The fibrin gel is used to keep the cells in place. And after doing so, we apply a topical tobramycin for disinfection. So this is the shadow wound on the cornea, and it's a little dip in it, as you see. And this is after we apply the cells and fibrin gel, and you can still see the wound is right here. So at day 7 post-surgery, we put the mice under anesthesia and we put them under the microscope to analyze their eyes. After doing so, we do an anterior segment optical coherence tomography, which is an ASOCT. So we do that to examine the cross-sectional corneal features. So this is the mouse holder, and we place the mice right here, and then we basically have to crank this in order to angle the mouse's eye into the detection probe. After doing so, we use the ASOCT bioprint to take a scan. And this is what the scans will look like right here on the bottom. So at week 2 post-surgery, we use the ASOCT to monitor the corneal changes. And then what we do is we sacrifice the mice by overdosing them with the anesthesia. So previous results showed that corneal stromal stem cells, CSSC treatment suppress corneal opacity formation, whereas the untreated wound control develops intense scarring. So right here is the wound and the wound control, and this is the stem cell treatments. As you see, the stem cell treatments have a smaller scar region, and each of them had different types of improvement compared to the wound. So I was given 400 different slices, 400 different scans of the cornea, and I basically had to put it into the Fiji app, and I put it into a 3D viewer. This is what it looks like after it's put into the 3D viewer. As you can see, there's unrelated light scattering debris on it, and I basically just had to cut it out, which is shown right here. So after the cornea is smooth, I put it into Fiji again, and I get a heat map. A heat map will show the different types of thicknesses in the cornea. So after doing so, I'm able to plug it into Metamorph and get the measurement. So this is me getting the measurement, and this is just a picture of what the cornea looks like on the bottom. So the cornea isn't like flat, it's quite rounded on the bottom, and then this is another snapshot of what it looks like on top. So here's a graph. So each dot in the graph represents a different cornea. So we have about 45 corneas per treatment, and each treatment represents a different donor cornea that was given to us. So as you can see, the wounding showed a higher volume than those in the stem cell treatment. And the stem cell treatment cornea had a reduced stromal volume compared to the wound control. So reduced stromal volume is the same thing as saying that the cornea is thinner. In this graph, we had the wound control at 100% for normalization. As you see, the stem cells are all lower than 100%, which basically shows that the volume changes decreased. So we put all the data into a Mann-Whitney U-test in order to calculate the p-value. So a p-value that is under 0.05 is considered to be significantly different compared to the wound. So these three treatments specifically were significantly different when compared to the wound controls, whereas these two weren't significantly different. So 3D OCT reconstruction allowed a precise measurement of corneal stromal changes, and after stem cell treatment, the reduced volume illustrated a recovery from fibrosis-related changes due to injury. So the volume analysis could be a potential parameter to evaluate the different effectiveness in corneal wound management. So the 3D OCT assessment work is still ongoing in my lab, and my bench supervisor, Maura, is working on scar volume and density measurements. More corneal samples will be analyzed, and I'll be participating in helping analyze them even after the program ends. So the lab develops appropriate cell therapies for corneal scarring in order to reduce limitation on the supply of corneal tissue, and to improve a scarless corneal wound healing, and to retrieve eyesight for patients with corneal blindness. So this is a couple pictures from when we were in the lab. This is my mentor, Dr. Gary Yam, and he was performing the algebra ablation on the mice's eye. This is me and Maura. She placed the mouse into the holder, and I was able to take an ASACT image of it. So I spent a couple days in the clinic, and it was a special experience because I was able to see my mentor, Dr. Vishal Janji, examine people's eyes, and I was able to see how different issues in the eyes were diagnosed and treated. So this is just him doing different eye exams. So great appreciation to my lab mentors, Maura Gary and Dr. Gary Yam, the support of Kira Lathrop for the image analysis, Dr. Vishal Janji from the Cornea Clinic, and my lab mates, Dr. Matin Santra and Dr. Tan Bing Yang. So also special thanks to Dr. Yuanyuan Chen for organizing the summer event, and Professor Jose A. Sahel for a great opportunity in working in the ophthalmology laboratory. Many thanks to Dr. David Boone, Stephen Jones, Solomon Lipschitz, and the Hillman Cancer Center. Thank you.
Questions? So let me ask you a question. What do you feel the most enjoyable moments and most challenging moments in the summer?
I would say the most challenging moment was sitting in front of the computer, analyzing all those images, because though it was kind of easy for me to do, it's just a lot of different samples and it's kind of a repetitive process. I really enjoyed going to the clinic because when I'm older, I know I want to be in the clinic. So it was just good to see how it works and see how you diagnose and treat different things.
Does this unique experience have any impact on your future direction? So your career?
I'm just honestly, right now, I'm trying to bounce around to see what I like and what I don't like. I did like this, but I don't think that this is what I would want to do when I'm older. Though it was a very good experience, and I appreciate all three of my mentors.
Yeah, it's very hard to find a short project for you to finish and make something that's presentable. Yeah, so I think for both of us, this is a real challenge for us to do something in a short time and to show you how it works, and also you have to learn something new for you, and also you have to do the analysis and also run up the project. But you have to do very hard.
The next speaker. The second speaker is Jasmine Horton from Dr. Patrick's lab.
Thank you. Do you want me to talk a little bit? So we can...
Yeah.
Okay.
Wait, just stand in the camera.
Stand in the camera. Where's the camera over here?
Yes.
There we go.
Can you help her click slideshow after your intro?
Sure, sure, sure.
It's a little bit hard. Yeah.
That should be okay. Not much your computer. How's that? Yeah, I don't have a ton to say. Jasmine's been great in the lab. I think for myself, it's an opportunity. I know we've talked about what we can provide to the trainees, but I think they provide some inspiration to us, and we all start off somewhere. So it's a good reminder of what got us interested in what we're doing in the first place. I think the challenges of coming up with a short project in a short amount of time are difficult, but we came up with a few things, and Jasmine was a willing trooper to do. You're going to see the fun parts today of playing video games, but watching dots move back and forth left and right on the screen for hundreds of trials are the less exciting parts, and she did those willingly too. But those are less fun to go through. Yeah, Jasmine was great. I feel like she's part of the lab. I think we'll miss her. She's a great personality. She's very smart, asked a lot of questions, jumped right in. So it was a much better experience than I even imagined. So she's always welcome to come back and check in on things, play video games, track her eye movements. I think she'll talk to you today about eye movements and learning. So there is a point to playing the video games. And I also want to acknowledge Kendra, my graduate student, and Sean in that corner there, who also spent a lot of time and energy with Kendra too. I mean, with Jasmine too, talking about different aspects of her experiments. We have a whole whiteboard now filled with multiple years of experiments, which we'll probably never get to, but if we get to them now, we know who to contact. So thank you, Jasmine, and good luck.
Yeah, let's take a picture.
Okay.
Mask on, mask off. Mask on one,
We'll do one of
and
each.
mask off one. You continue mask off. Patrick, maybe move around. If you are really.
My face is blue.
Yeah.
How's that? Okay.
Cannot find my.
There you go.
Oh, there we go. Slideshow.
Can you usually do that?
Oh.
There you go. Perfect. Okay.
Take your time.
Yes, what do you do?
Hi, everyone. I'm Jasmine, and Dr. Mayo was my mentor. And over the summer, we studied using eye tracking to understand how experience impacts video game performance. So the most important way we learn, or the most common way we learn, is through visually processing our surroundings, which is also gaining experience. And experience is observation, or to encounter things. Like I can gain experience writing by just holding a pencil up to paper, you know? Anyway, experience generally enhances performance. And with experience and time, your eye movements can change. We are learning through this, well, we're learning through the summer how experience shapes eye patterns. This was our setup Excitebike 64. Over there was the headrest that we used, the TV. It was fun to set that up. We used the Nintendo 64 with the weird looking controller. And here's what Excitebike looks like. We didn't use this track. We had our own track that we made, but I don't have a picture of it. Our hypothesis was that as the number of laps completed increases, lap time will decrease and eye movement variability will shift from the biker to oncoming obstacles. So first, we recorded everything through Tobii Glasses 3. We spent 2 days on this. I did 10 games per day, 5 games before lunch, 5 games after lunch. The glasses had a recording rate of 100 hertz or samples per second. We used MATLAB to plot all the data from the glasses, and we had to do a lot of math and a little bit of coding to try to figure out how to get the desired data from the recording, because it was a problem that all the graphs that we originally came up from the raw recording was too much, because it was the whole video and we just wanted the 3-minute clip of me playing the game. So we extracted the gaze data, which was the data from the goggles, and we were able to get graphs that were from specific parts that we were looking for. This is a graph that I made of the lap time versus race number and the crashes versus race number. Day 1, I kind of didn't really know what I was doing. Excitebike was a brand new game for me. I didn't really know the controls, so I was crashing left and right. You can see there's a general downward trend, a higher learning curve of having less crashes on day 1 than on day 2. One thing I found interesting was you can look in 9. Here I have about 6 crashes, but my playing time was about 3 minutes, 30 seconds, which is really high. This was because I crashed into a wall for about 30 seconds. Yeah, it was kind of embarrassing. So day 2, you can see that the crashes kind of corresponds better with the playing time. Higher crashes, higher playing time. I was able to actually recover. I did get stuck in a wall, but instead of for 30 seconds, it was actually only for like 5. So I feel like that was improvement on my part. All right, so this is the data from day 1, the slowest game, game 2, and the fastest game, game 6. It's like a 50-second difference. You can see the eye movements of the first 30 seconds of the game in game 2 are very all over the place, very distracted. I was kind of learning the game out, and I wasn't really sure what was going on. I was like, oh, I want to look over here. No, let's look over here. Let's look over there. But then by the time I got to game 6, it was more controlled, and I was looking more at the player. I was looking ahead of the player, looking on what road I was going to next. You can see the variance, which is how much the eye movement changes on the X and Y axis from the average. The variance decreases from game 2 to game 1 on the X axis and also on the Y axis. So numerically, I'm physically looking at a smaller space on the latter day. Do I have to press a button? Okay. Here we go. So these are videos from the days that I took. It's all the same turn. Let's see if I can click it. Did I do something wrong? No. Okay. The mouse is stuck on the bottom screen. Don't mind me. I feel like I should be better with technology at this point.
Yeah, is it not moving? Is it not playing? Okay. Wait a sec. Let me find your original one. Hopefully. You are converting from Google Slide, right?
Yeah, it was from Google Slides.
Sorry, technical break.
Anyone have any good jokes?
So I have a question, a quiz. Among 3 sports, which one has the highest eye injury? Baseball, basketball, and football.
Basketball.
Why you know that?
Fingers.
Yeah, good.
See. Okay, have your email here. Let's figure out what's going on. Jasmine. Let's see if that one works. So. Around. Slideshow. Okay, I don't have access. Quest access.
I think we need to be logged into as we have access.
I'm sure. Make sure that the videos were. This should be assigned to quick Google Slides for me, but my computer can't with PowerPoint.
My. Oh. I lose track. Oh, here you are. Or the easier way is to log in your own Google account, maybe. Yeah, you can log on. Yeah, just log me off.
Now we can switch the button then to use this computer. Try to log.
Yes, I'm.
Okay, okay, yeah, that's still.
Oopsies. GMAIL. This is the one day that he's not supposed to go right. Do you want me to click allow and allow? That's good. Why not? What? Can I minimum X out of this? By X out, will it cancel on my progress? And then I can just hit slideshow. Okay, here we go. Yay! Okay, so this is the same turn for days 1, games 2, and game 6. You can see here, I'm kind of looking all over the place, and I don't really know what's going on. And then you can look at game 6, which is quite the improvement. You can see I'm looking ahead up until I crash.
Can you repeat that again?
Yeah. Here, day 1, I'm all over the place looking at the speed. For some reason, I look way over up to the top. And then game 6, it's just very calm, very nice, more focused on the game. Again, up until I crash, then I just start looking at him. Okay, so this is day 2. The TV was a little bit closer on this day because we could get better graphs. Here, I'm also kind of looking all over the place. It was a slower game compared to day 2, which was faster. You can kind of see I'm looking a little bit higher on the screen, which was noticeable in game 6 on day 1. And then you can see, even though I have these little loops here, these are just quick 1-second eye movements, and it's about 4 of them I made to that direction. But then over here, you can kind of see it looks a little mess. I like to say that this looks kind of like a pterodactyl. Like you can see the little teeth and the little wings. It's a little messy. You can also see the bars, the variance, the X variance, surprisingly, gets higher on day 2, which is kind of supported because you can see a lot more of the X-axis than the game 1 is taking up. But the Y variance shrinks a little bit, just a little bit, from the 0.0208 to the 0.0200. And here I have the games. It's the same turn as is day 1. You can see I'm looking ahead until I start looking, and then I crash when I look at the guy. And then here, it's almost a perfect turn. I'm pretty proud of this one. I'm looking ahead the whole time, and then I go into the little bumps and it's all smooth. I'm so proud of it. Here, I can show it one more time. The difference. You can see I'm looking everywhere. And then I go off the side, and then it's smooth sailing. I think it's very satisfying. Okay. In conclusion, the eye movements during Excitebike were more focused and concentrated over time and experience, and it was in function with the general downward trend of time. Further directions, we could improve performance of other areas by figuring out how to train desired eye movements. Like instead of looking at object A, we want you to look at object B at this time. And it could improve learning processes, and it could maybe make rehabilitation or treatments easier. If I were to do this project longer, I would probably change different variables. Like you can change the dirt type track. You can change if it's night or day. You can change what rider you do, so you can drive like faster or slower and be more agile. And maybe I would see how distractions affect eye movements. Like if a visual distraction all the way in the corner, not on the screen, makes you have different eye movements than like a sound distraction, like someone's walking behind you. Or maybe if you're just hungry, how does that affect you? And this is my acknowledgements. I would like to thank my mentor, Dr. Mayo, Dr. Sean Willett, and Kendra Nunman, because they were here to help me, especially with MATLAB, because I had no idea what I was doing. Dr. Yuanyuan, Chen, and the ophthalmology site. This was a lot of fun this summer, and I would like to thank everyone who was a part of the Hillman Cancer Center.
So were all the other students jealous that you got to play video games on?
Yes.
I like to think I was very lucky to have a mentor.
So I have a question. If you compare your first experiment on your day 8 compared to your day 1, do you still see improvement?
Yeah, I see improvement because if you look at it at time-wise, game one of day 1 was very slow. I was crashing left and right. I couldn't figure out how to turn, and I didn't know where to look. I, at that point, didn't know that the speed area was important to actually look at. And I was just looking at plays trying to get to first, and I was on the amateur level. I was on like the easiest of the easy levels, getting last place every time. But then on day 2, whenever I was better, I was able to get first place once. I was very proud of that. And everything was easier for me. I was able to turn. I was able to speed up when I wanted to speed up. I felt like I had more control over the game at that point.
So another question, when you are really, really good at the game, do you think your eye will have more distracted movement because maybe you gain confidence, so you have extra energy that you can move around your eyes as you still can control the car?
I think that when you get really good at the game and you're very comfortable with the track and you're able to just muscle memory everything, there might be more distraction because you won't have to focus so much on the game because you'll know what you're doing. I didn't really know what I was doing, so I had to look at the screen to pay attention. Maybe one day if I got more comfortable with Excitebike, I would be able to do that. Oh, I can look away from the screen for a couple seconds, but I'm not near there. Any other questions? Thank you.
Next one is Marcus, right?
So our next speaker is Mr. Marcus Jones. So he's from Central Casting High School, right? And he was doing great in my lab. So you know my lab is, so the primary focus of my lab is to develop cutting-edge OCT systems. And then right now we are developing or building a new visible light OCT system for high resolution retinal imaging and also some function images. And so the building a system requires a lot of alignment for the light beams. Sometimes the alignment can be achieved by some commercially available components, but most of the time we need to build or design components our own and then 3D print them to fit for the purpose. And Marcus, so Marcus built the computer at the first, and then learn the software, and then set up our 3D printer. And yeah, he is really doing very good at this. And so let's welcome him and check out how he achieved that amazing.
She was taking with and without the mask. So let's start with mask first. Okay, right. Strip.
Make sure I don't trip here. Hello, everybody. I'm Marcus Jones. I go to Central Catholic High School. My mentor is Dr. Pai. And my project was 3D printing components for an OCT prototype. So a little bit of background here, the anatomy of the eye. So with OCT, it focuses on retinal imaging. So the retina is responsible for receiving light that comes in from the lens here. It contains all photosensitive cones responsible for your vision. However, some things can happen, like diseases. So how do we diagnose those? With OCT. OCT is an imaging technique used to take scans of the retina to make sure everything is okay. It's used in the medical field to help diagnose these problems. So where does my project come in? So my project involved designing these 3D components here for this OCT system that we are building. The main objective was to create these custom pieces so we could better align the light in the system so it can go from component to lens to camera to make sure everything is working properly. And this is ultimately going to help a patient be diagnosed with any retinal diseases that they may have. So the first thing I had to do was set up my computer because I'd be using this all 7 weeks of this program to help me design these pieces. And it took about 2 days to build. I had to install everything like SolidWorks and then PowerPoint to do this presentation, for example. And yeah, I'd be using this computer to design all my 3D components. So a little bit more about SolidWorks. SolidWorks is where I'd be spending most of my time. It's a 3D modeling software, and it provides things like sketch tools to draw lines, circles, or fillets, which are just smooth corners. These right up here are the sketch tools. You got your lines, circles, fillets, everything like that. You could also extrude and cut designs. So for example, right here is my first project that I was working on. So this, these blue lines here are the sketch that I made. Then I would extrude it, which is making it into a 3D model like this down here. And then with the circles and everything, you would use extruded cuts to cut into the 3D model. So you would have a hole. However, this was not smooth. It did come with some failures. Here's an earlier prototype of my first design. For example, it was too big, so it took up too much space. The holes were misaligned, and the holes weren't deep enough. So the purpose of this was to mount a camera on top, and then these holes on the outside would be used to screw in the piece into a breadboard, which is just a big board with a lot of holes you can use to screw things in. But yeah, it was meant to put a lens here, screw in here, and screw it into the breadboard so it can be used to align light. But with those problems, it couldn't do any of those. So my second project here is a lot more complex, as you can see. The purpose of this was to align two components, one called a kilometer and a grating, to diffract light into another component we were going to build over here. But these 12 holes on the outside used just like the other one to screw into the breadboard to keep it in place. And these two, so the kilometer would go here and the grating would go here, and light would pass through here and be diffracted over here. But like I said, it didn't go smooth, just like the first one. Some failures I had, the holes on the extruded parts, which are the mounts here for the grating and kilometer, were not correctly centered. So if we placed it here, we couldn't screw it in because the hole was not aligned properly. The distance between these two was not proper, as well as the angle, which is very important when you're dealing with light. It has to be diffracted in a certain way to get to the next component that we need. But yeah, all these failures I had or misjudgments, I couldn't do that. But after all the everything I fixed, after I fixed everything, we took it to our 3D printer. This is our 3D printer. So first, in order to 3D print, you take the file from SolidWorks, convert it or export it as an STL file, and then you put it into this slicing program, which is used to 3D print. It gets converted to a G code, which a G code is just, think of it as like a blueprint for the 3D printer. It's like the only file the 3D printer can recognize and use. Because when a 3D printer is printing, it's printing a 3D image from on top. So it has to have that blueprint on what to do. And the software we had allowed us to customize various things like the printer speed, material supports, etc. So here is an image within that program, the slicing program. So there's four main things I want to highlight here. First would be the material. That would be the material you're using to print this model, whatever you want to print. The second one would be the layer height. So when a 3D printer prints, it prints layers upon layers. And the layer height is just how tall that layer is, essentially. And the other two is the infill density, which is essentially when a 3D printer is printing, it's essentially how much between 2 points or 2 lines that it fills in. And then the infill pattern, each layer it's selected as zigzag. So each layer is printed, let's say one layer goes this way, another goes this way, another goes this way, like a zigzag pattern almost. But here's one component in that slicing program. This was the second project I had. And then eventually, after we sent it to the 3D printer, waited some time, then here it is printed. So here's some pictures that we took after all the failures were accounted for and we got it printed and working properly. So like I said before, this is the second one. Kilometer goes here, gradient goes here, light comes through here. Some light doesn't get diffracted, it goes straight like this here. And then the other light gets diffracted this way, which would be needed to diffract it to this lens here. This is just a demonstration, so it's not fully finished here, but light goes through here, like I said, through the gradient, diffracted this way, it gets bent into like a line almost. And this is the entire system that we had a few weeks ago. So the light would be produced through here. Some light would go into the sample here, which is where you get your samples, like retina samples from people. This is a reference. And then the light would go all the way down to here, which is those two. And eventually onto a camera. Hopefully this video plays, but this is just a demonstration here on the distance had to be correct. So right there is a sweet spot on where the camera had to be placed in order for the light to be proper. Let me see. Play that again. So it's blurry from a distance. You get closer, sweet spot there, closer and closer, blurry. So the camera had to be placed in the proper position. So we would have to make something to keep the camera in place there. Okay, so whoops. At the time of this presentation, this is what everything looks like. Same thing, but we added the camera like I was talking about. So right here, you can actually see the light that got diffracted from the lens into the camera there. So we positioned it in the sweet spot there. And then after, oh yes. So I actually brought a couple of those components so you can see them. First, I'll show them on here, Zoom. So this is the first one. Just making sure they see. Second one. And pass these around. So that's the second one. Those are the two I work on. So, you know. But yeah. Then as a follow-up, after all is said and done and that OCT is built and scans get taken from it, we loaded into a program called MATLAB, which is like a programming thing. And so you put the scans in here. So here's the scans of the retina. And then an algorithm is run to separate these layers so they're easily recognizable. So each line here is a different retinal layer. However, the algorithm is not perfect sometimes. It gets messed up. So somebody has to go in there and manually fix them on different frames. So that's kind of what I did for a little bit of time, just to understand everything I was doing after the OCT prototype was made and everything. Then that's eventually where that would get to. Then my acknowledgements. I'd like to thank my mentor for all of his help, everything, teaching me. And Pitt and everybody at the Hillman Academy for giving me this opportunity. Everybody at the Eye and Ear Foundation, the ophthalmology site. It was such a great experience, so much fun being here. And yeah, that's it.
Any questions?
I always wondered what happens to the 3D, what happens to the plastic on the ones that you misprint for the 3D printer? Can you reuse that plastic?
I'm not so sure, but like I kept some of the failure prints.
It's like a
Just-
memento.
Yeah, just as a, and it's also helpful for seeing what I did wrong because sometimes on the computer you don't recognize it. But yeah, I'm not so sure if they can be reused, but I just use it as a learning experience, really.
Makes sense.
Can you select material for your 3D printing base? Like, is it only one type of material you can use or you can choose?
So you can choose. For my components, I only used one type, but I believe there's like 7 different types or something like that. I didn't really look into that. But yeah, the printer we got can print carbon fiber, so it's pretty cool. Yeah, anything else?
How long would it take?
It depends on the size and everything like that. So like for this, my second project, it took roughly 5-6 hours maybe. And for something like this, maybe about 2 hours. It really depended on how it was shaped, how many complex things, how much material you're using, pretty much. But you could go in there and adjust the printing speed. However, I didn't do that because that you would risk accuracy. So it wouldn't come out right, but we needed it to be precise, so.
Actually, Max has designed some other models. I feel
Oh,
different,
yeah, I guess pass this box around.
yeah.
Those are all the models. I didn't print all of them, but those are all the models that we used at some point or failures. But those are parts that are not needed anymore.
Did you design anything else, like a toy dinosaur when he wasn't watching?
I did, he did let me print out a certain design, whatever I wanted. So I just made a little thing for my mom. So,
Did you have experience in 3D printing before?
last year I took a CAD class, which was something similar to this, but we were given the dimensions on everything. But this, you just kind of had to figure it out and do it on your own. I mean, I did have a lot of help from my mentor, but it was a lot different from the class. So, yeah.
You know, Dr. Sahara chair has come and I'm really thankful for the support and he's traveling back and forth from downtown here just to say hi to everybody.
Yeah, we have to take a mission of mercy, so we are providing free care to thousands of people, dental, ophthalmic, and respiratory care. So we have a big operation today and tomorrow at the convention center.
So I will propose like all the scholars in there and we can take picture with our chair. And then I'll send everything. And here's the high school teacher. Like a teacher.
Thank you. It's a sign of COVID.
Okay.
Great. Welcome. Nice work.
Dr.
Oh, sorry.
Dr. Chen, he's putting that cord up there. It's giving me a heart attack question.
I know. I'm so sorry for harassment. No hazard. Okay. Oh, Marcus. Stand there with Dr. King.
I took a picture. I can give it to you.
Oh, yeah, all right. So next one is Jonathan. No, is that? Oh, it's Daniel, right? Daniel. Yeah. Next one is Daniel. Dr. Shank, go
Should I
ahead.
start? Okay, so it's my pleasure to introduce Daniel Komlosi today, who worked in my lab for the last 6 weeks or so. He approached me maybe 2 months ago. He'd been working with Dr. Kinchington there in the back, and he was one of the only students I've ever met who actually came with an idea beforehand. So he came on an idea, and we ended up changing it a little bit, but he ended up working on his idea. So it was really driven by his concept. And he did a fantastic job. He was extremely enthusiastic and curious about science, sometimes too curious. And we'd end up talking for a couple hours every morning about ideas and things like that. But he did a fantastic job. And today he'll be speaking on evaluating a novel GFP tag in the yeast Saccharomyces cerevisiae. So take it away.
Yeah. So yes, my name is Daniel Komlosi, as Dr. Shanks has so kindly introduced me. And for the past 6 weeks, I've worked in his lab working on this project. So evaluating a novel GFP, which stands for green fluorescent protein tag, in the yeast species Saccharomyces cerevisiae. Okay, so a little bit about the goal of the project. So the goal of this study was to test if this newly discovered approximately 20 amino acid tag, N-terminal tag, which I will refer to as 580N throughout the presentation, would increase the brightness of GFP in Saccharomyces cerevisiae compared to GFP without the tag. So you can kind of think about this as like a cage fight here. You have the GFP by itself, and you have the GFP with the 580N tag right here. And for the study, we chose a specific type of GFP, which is made to be expressed in yeast called MUKG1. This was based off of literature that we had read prior to doing the experiments. And then we hypothesized that the GFP with the tag will be much brighter than the GFP only, and I'll get into why that is. So a little bit of background about my project. So I wasn't the first one to test this amino acid tag. It was actually one of Dr. Shanks's colleagues who came up with this or discovered this tag and tested it in several bacterial species beforehand. The images depict the surface of an agar plate, and this is Klebsiella pneumoniae. You can see that there are 3 patches of bacteria. The first patch here shows the GFP by itself. The second patch shows the tag we're testing with the GFP. And then this third final patch is just the bacteria without any GFP or without the tag. So this picture right here is when you excite it with ultraviolet light, and I'll get into what that means. In the same GFP and the same patches of bacteria here, but you can see that with the GFP, there's minimal fluorescence intensity. And then with the no GFP and no tag, there's no fluorescence at all, which is expected. But then surprisingly, with the tag and the GFP, the brightness was significantly brighter, or it was significantly more bright. So from this, we can conclude that in bacteria, this tag does make a huge difference in terms of brightness. Okay, so what exactly is GFP and why is it important? Why am I talking about this? So when I say GFP, I actually mean green fluorescent protein. It's just the acronym for it. And green fluorescent proteins were first isolated by a man named Osamu Shimomura in 1962. It was first isolated in the jellyfish Aquaria Victoria, as shown right here, in the top right. And GFP functions just like any other protein. It could be expressed in any living organism that includes yeast, bacteria, and mammalian cells. It isn't naturally found in all of those cells, but it can be expressed in all types of organisms. Now, the very interesting quality about GFP and what makes it GFP is that it can be excited using ultraviolet light, as I mentioned before. So this means that you would shine that specific wavelength of light onto the yeast colonies or the whatever living organism that express that gene or that protein, and it will appear green to our human eyes. So this makes it a very useful tool in molecular biology. Now I'll get into some examples. So GFP allows proteins to be tracked within cells, and this is called localization. It can also be used as a recorder to measure promoter strength. And it could also be used to enhance the visualization of cells. So very useful tool in molecular biology. There are also many different types of GFP variants that have been discovered after this discovery in 1962 that have been designed specifically to improve the function of GFP, to make it brighter and more stable. And we are using the GFP MUKG1, which actually functions exactly like GFP, but it was originally isolated in soft coral, not the jellyfish. Oops. Okay, so what is Saccharomyces cerevisiae and why am I using this? So Saccharomyces cerevisiae is a species of yeast, very widespread and has been known for centuries, and is a very commonly referred to as baker's yeast because of it being known for fermentation, so winemaking, baking, and brewing. And it's a very widespread model organism because it's a very simple eukaryote. So that means that they share a lot of the basic biological properties with our mammalian cells. And also genetic manipulation is very easy and cheap. It also grows relatively quickly. Okay, so what exactly are plasmids and how are we using them as scientists? So plasmids are these genetic structures found within cells that is completely independent from the main genome of the cell. Like for example, we have a bacteria here, and this is the chromosomal DNA here, and we have this independent plasmid. We have the independent plasmids right here. And they're typically these small circular DNA pieces. Okay, so we have to wonder how are scientists using them? Now, scientists use them as like little tools to express their proteins and genes of interest. So for our study, we want to express GFP within yeast. So that's why we use this plasmid to express it. And I'll be saying this word vector within my slideshow. And it's just essentially, think of it as a plasmid that researchers use. So just think of it as a plasmid. Okay, so methods using this technique called homologous recombination, which is specific to yeast. We were able to generate two different plasmids or vectors, one with the amino acid tag right here with the GFP and one without it, so just the GFP. These were generated. This represents homologous recombination. In addition, we also had a plasmid or vector only without any GFP or without the tag. And for all of our plasmids, we used a strong constitutive promoter called ADH1. And in order to verify our work, to make sure that the plasmids were correct, we used PCR verification. And in order to quantify our data, we used a dissecting scope to take images, and then we processed those images in a software called ImageJ. In addition to that, we also quantify our data using a fluorometer. So results, these are pictures taken of the surface of an agar plate. So you can see that there are some healthy yeast colonies here, all growing. And this is the, these two images show the GFP with the tag that we're testing. And these bottom two images show just the GFP only. So you can clearly see that whenever because these images are, bless you, these two images are when we excite it with the ultraviolet light, and you can clearly see a difference in fluorescence intensity. So you can see that when the tag is added onto the GFP, there's a lot less brightness compared to just the GFP alone. So this is a qualitative look at our data. We obviously have to quantify this as well. So we did this with a fluorometer. So there's a picture of fluorometer right there. So these are our 3 plasmids that we tested. And RFU stands for relative fluorescence unit, which essentially just takes the brightness or the fluorescence intensity and divides it by the yeast cell concentration. So you can see that for our plasmid only or vector only, with no GFP and no tag, there was 0 fluorescence as expected. But when you compare the tag with the GFP to the GFP by itself, you can clearly see a difference in GFP, or sorry, in fluorescence. And our p-value was less than 0.05, which means that there is a significant difference. So from this, we can conclude that the 580N tag showed no increase in MEKG1 fluorescence intensity in the yeast species Saccharomyces cerevisiae. Therefore, our hypothesis was wrong. But some possible future directions, we can sequence the plasmids to really make sure that the genetic makeup of our plasmids was proper and we didn't mess anything up along the way. And we can also express MEKG1 in other types of yeast species, so specifically infectious types. And I would like to thank all these people, and specifically the Hillman's Cancer Center Academy, as they have been provided me this amazing opportunity to work in a lab setting, and obviously Dr. Chen, our site head, for being so kind and providing us with food every Thursday. And I'd like to thank Dr. Kinchington for setting me up with this program, and obviously Dr. Shanks for letting me use his lab, and Nick, Nicholas, Stella, and obviously Rachel Calvario. So thank you very much.
Questions? So I have a question.
Sure, go ahead.
For this 58N, you have found that the green fluorescence being decreased.
That's
Have
right.
you look at the possibility that there is a chromatic shift, which means maybe the optimized absorption and excitation may shift elsewhere?
I'm sorry, so
there is a theory in the protein. When you change the amino acid, the microenvironment within this chromophore may shift is optimized absorption and excitation.
yes.
So maybe what you have seen with reduced fluorescence means that the peak is no longer at where you're checking. Maybe you should check other wavelengths.
Yeah, that would definitely be a possible future direction we could test. Yeah, I know that the 580N tag, it essentially stabilizes the GFP. So yeah, that would definitely be something that would be a good idea to try. Yeah. Thank you so much. Yeah.
So how could you do that? Is there like a scanning microscope or something like that?
I don't know if you can answer that.
Yeah.
So yeah, we have spectrophotometers that allow us to style it exactly the way it might. In this case, for him, we used a filter-based one, which kind of had a broad range. So I don't expect it would be outside of that range, but it is definitely feasibility that you're on.
I was just wondering, are there other tags that you focused in 580N? Is there anything else that you might want to try?
Other types of tags?
Yeah.
I'm sure there are others, but we mainly focused on this one, this specific type, because Dr. Shanks did approach me with this idea, and I was really interested. But definitely comparing it to other types of tags would definitely be a good idea. Yeah.
What were some like highlights and lowlights of the project?
Oh yeah. Well, I really enjoyed working with the lab members, Nick and Rachel, and obviously Dr. Shanks. I really like the pipetting and showing them the techniques that they use to work with the DNA and the plasmids and the yeast and culturing them. It was all really fun to work with that. So yeah, I really appreciate that.
So this is one question. So this is because of the different folding in eukaryotes and bacterial cells is creating this difference?
Yes.
Certainly.
Yes, exactly. So that could be one reason. We're not exactly sure, but there's it could definitely be the protein folding. Yes. Yeah.
I guess one other possibility is that Daniel selected a monomeric version of the GFP type of molecule. It could be that this tag is facilitating dimerization, as what happens in a normal GFP. And in this case, it doesn't help it to become a dimer. So it actually might inhibit its folding function. Thanks.
Oh.
Can you both shift out of this right now? The food is coming.
Now smile for the food. Do you know who's next? Yeah. Oh, yeah. I've lost the. Oh, there it is. Jeez. Jonathan, right? Yeah. Now, do you know how to activate this? Yeah, I'm not sure how to switch to the full screen. I mean, normally you press, there's something down here, but I can't see it.
I don't usually use PowerPoint.
I think
this thing's blocking it.
yes. Yeah, I can see that. Dr. Do you know how to turn this into open full screen mode? The cursor's up here. Ah, there we go. Here's Dr. Chen to the rescue.
Right. What's happening?
I don't know. We double clicked it. and it
You
showed
double
up
click,
here.
oh, it's so there.
Oh,
Oh, yeah,
oh.
you need to drag this.
Yeah.
Oh.
The food, one of the food is being canceled again. I don't know what happened. I don't know. A lot of stress.
Just to drag it from.
I guess they have it under control.
Yeah.
Okay. Thank you. Slideshow. So our next speaker is Jonathan Lee, and Jonathan has been born in a small town in Illinois, and he was living with his grandma in the first 3 years, so he had really fluent Mandarin speaking. And then at year of 3, Jonathan moved to Pittsburgh and become a Pittsburgher. He spent the entire education phase at Fox Chapel school area. And it looks like he's doing very well scholarly. And through the short period of time I was being with him, I learned he's the vice president of the math club, and he's also painting really well, have some
state-of-the-art on painting. So, and also from science part, I found this is also one of the smart kid that I have ever met, also including all of you guys. And I have to say, I was really impressed by every one of you for your really funny and very smart questions. And I enjoyed a lot working with all of you. And today, Jonathan is gonna talk about a small project for genotyping PCR using the animal model of retinitis pigmentosa. This is a small project, but this is one of the founding method for any animal models that if any of you are gonna use in the future that are genetically modified. So, Jonathan.
Okay, master off picture.
We can take it.
Yeah.
Later. Yeah. Okay, thank you.
Okay, hello, my name is Jonathan Lee, and this is my helmet project on genotyping PCR for animal models of retinitis pigmentosa. Okay, so before we talk about retinitis pigmentosa, we have to first understand the eye and how the retina works. So this is a diagram of the eye. And so whenever light comes in through the lens, it hits this backward C, this purple backward C, and that's the retina. So here's a zoomed in picture. So when photons of light come in and they hit the retina, this layer, which is called the outer nuclear layer, which is made up of photoreceptors. So these grayish looking baseball bats, the photons hit the baseball bats and they send messages through the neural system to the brain so you know what you're looking at. Okay, so now that we understand how the retina works, we can look at retinitis pigmentosa, which is a hereditary disease that causes blindness over time in the eye. So here's a picture of a normal retina, and this is a picture of a retina that's been diseased by retinitis pigmentosa. So you can see these black spots around. Those are the dead photoreceptors, and that causes vision loss. So here's normal vision, and after you have retinitis pigmentosa, your peripheral vision begins to degrade, and you eventually are left with tunnel vision, and most of the time you get to blindness. So there's currently no cure. So because retinitis pigmentosa is a hereditary disease, we have to understand some basic genetics to understand how the disease is passed down from person to person. So everybody has genes. For each gene, each person has 2 alleles, one from your dad and one from your mom. So there are dominant alleles and there are recessive alleles. So dominant alleles mean that if you have one of the dominant alleles, at least one of them, then you get the trait that comes with the allele. But you need 2 recessive alleles to get that trait. So 2 different alleles would be heterozygous, and then 2 of the same allele would be homozygous. So for example, we can use this eye example. So there's the brown eye allele, which is dominant, and then there's the blue eye allele, which is recessive. So if a person had a brown eye allele and a brown eye allele, it would be homozygous, and they would have the brown eye trait. If they have a brown eye allele and a blue eye allele, they would have brown eyes with heterozygous. And the only way for them to have blue eyes is to have 2 blue eye alleles because it is a recessive allele. So now that we understand some basic genetics, we can see how that applies to retinitis pigmentosa. So there's 3 ways you can inherit retinitis pigmentosa. There's autosomal dominant, which is where only one of your alleles of your gene is mutated to cause the disease. There's autosomal recessive, which is where 2 alleles are mutated. And there's the X-linked, where a gene in the X chromosome is mutated and that causes the disease. So retinitis pigmentosa occurs 1 out of every 4,000 people. And the specific mutation that we're studying in the Chen lab is the P23H rhodopsin mutation, which accounts for 10% of all autosomal dominant retinitis pigmentosa. So to study this disease, we have to use an animal model to have test subjects. And we use mice because they reproduce at a very young age in great quantities very quickly. So at ages of from 1 month to 2 months, they can begin to reproduce. And they are pregnant for 4 weeks. And on average, the litter size would be 7 pups. So now that we have mice, we can give the mice the specific disease so that we can study them. So on the left is a picture of the P23H mutation being knocked in through homologous replacement. So basically, the gene that contains the disease is being marked so that we can identify it later on in the mice. And then the type of mice that we're trying to study are the P23H wild type heterozygote, which is, it has one allele that is the P23H mutation and one normal allele, so that it best reflects the disease in humans, because in humans, the disease is autosomal dominant, which means there's only one mutated allele. So to ensure all the test subjects are the right genotype, we have to breed a P23H homozygote with a wild type homozygote to ensure all the children are of the correct genotype. So now that we have the right test subjects for our tests, we can examine the mouse eyes to see how to see the retinal degeneration. So on the left is a graph comparing the outer nuclear layer thickness, which is previously mentioned, is the layer of the photoreceptors in the retina over time. So the top line with the squares is the wild type homozygote, which is a normal mouse, and then the rhodopsin P23H wild type is the heterozygote, and it's the one with the disease that we're studying. So that one has a lot steeper curve, and you can see that the outer nuclear layer thickness is decreasing, which means it is leading to blindness, because a thicker outer nuclear layer means healthy photoreceptors, whereas a thinner one means that they are dying. So in the second graph, you can see a P23H wild type heterozygote, and it's the outer nuclear layer thickness, but it's on different positions of the retina. So you have the peripheral on the outside, and you have the center. So we're comparing the control to the treatment. So the control is just a normal mouse with the disease, and then the treatment is a mouse with the disease, but it's also given a treatment over time. So at all of the positions, basically, the treatment is always at a thicker position, which means that the treatment is working because it's preserving the thickness of the retina, of the outer nuclear layer. So those were our results, or those were potential results, and we need to verify that those results are legitimate. So that's why we use polymerase chain reaction, or PCR, like Daniel did with his experiment. So we take a tissue from an animal and we add some DNA release additive, which is basically a solution that can break down the tissue of the mouse in order to release the DNA. And then we take out the DNA and we add a Taq polymerase, primer, DNTP, buffer solution, and water before we put it into the PCR machine. So the PCR machine is a machine that basically regulates the temperature of the mixture, and that facilitates the PCR process to happen. So say you have one piece of DNA and the PCR process starts. The first stage is denaturation, which is where the temperature goes up to 98 degrees for 20 seconds, where the DNA is double-stranded and it splits into its 2 strands at the higher temperature. And then comes the annealing stage, where the temperature goes down to 60 degrees for 30 seconds, to where the primers, there's a forward primer and a backwards primer, and they're able to attach to the split up strands of DNA to basically point out where the gene begins, the targeted gene that we want to duplicate begins. And then the temperature goes back up to 72 degrees for 60 seconds to the extension stage, where the Taq polymerase comes in and guides the DNTPs to the right placements to extend from the primers to create the new DNA. So you start off with one piece of DNA of the targeted gene, and then you go into, after the extension phase, you double that to 2. So that happens 35 times, and in the end, we get 2 to the 35th power minus 1 copies of the PCR products of the targeted gene that we're trying to look at. So now to determine the lengths of those genes, we use gel electrophoresis, which is a process where you take an agarose gel and then you put it into a gel electrophoresis machine. And the agarose gel has wells right here to where the PCR products are put into. And the gel electrophoresis machine has a negative end and it has a positive end. And then whenever the current runs through it, because DNA is negatively charged, it will move from the negative end to the positive end through holes in the agarose gel, which allow the DNA pieces to go through. The shorter bands will move faster, whereas the longer bands will move slower because the shorter bands will more easily fit through the holes. So it basically acts as a filter to filter out the short bands from the longer bands. And whenever we're running gel electrophoresis, we run it alongside a PCR DNA ladder, which is basically a solution that controls a lot of different different DNA with known lengths. So here are all the lengths of the DNA bands that are in the mixture, and then we basically use that as a reference to the lengths of the PCR products that we were running. So here are my results. To get my results, the agarose gel was pre-stained with a DNA dye so that we can use UV citation to visualize them. So this is my first experiment. This is my second experiment. And in both experiments, I had a non-template control. So this last column in each of the experiments. And that non-template control is basically used to verify that there's no contamination happen at all during the process. So in columns 1 through 4, a band showed up and they were all 400 base pairs. In columns 5 through 7, nothing showed up. And then in columns 8 through 10, each had 1 band and they were about 600 base pairs. So here's a reference for what the length should be for each mouse. So for a wild type mouse, the band should be 400 base pairs. So columns 1 through 4 of my experiment would be wild type mice. P23H wild type heterozygote would have a 400 base pair band and a 600 base pair band because it does have both alleles, both the mutation and the wild type allele, but I did not have any heterozygotes. And then the P23H homozygote would only have a 600 base pair band, which would be my columns 8 through 10. So conclusions and discussion. Through this process, I learned that PCR is a consistent and efficient method to identify the genotype of animal models. I obtained most of the results that I expected, and the 3 PCR reactions are probably due to the DNA sample being aged or the tissue sample being degraded, and that they will have to be retested in the future. So the confirmed mice will be used for efficacy tests in the Chen lab, and during my stay here at the Chen lab, I also shadowed many other experiments like iCup and rat dissection. So here are my acknowledgements. I want to thank Dr. Chen for being my mentor, Hillman Academy with David Boone, the OTP Roundtable seminars, and all the lecturers, Dr. Chen's lab for helping out, Chase, Abhishek, Nitika, Owen, and Riley, and the Irene Foundation and the Hillman Foundation. That's it.
Very nice talk. So Dr. Chen just went to grab some food because one of her orders was canceled again, so she had to go to a restaurant to grab something. Yeah, so that's very nice. I'm amazed by your talk and by all those kids. They are really smart. So then you not only do this and also just know a lot of this. So I just have one question about the failure of those 2 bands. You did not see any bands. So then you mentioned that maybe the tissue degradation.
Yeah.
Any other thoughts?
Any other what?
Other thoughts about
Thoughts?
other possibilities?
Thoughts about possibilities. Initially, some of the people in the Dr. Chen lab thought that the tissue, like we were extracting it and there wasn't much buffer liquid left into the tissue sample. So whenever we pipetted it out, we thought maybe we pipetted up some of the tissue and maybe that clogged up the gel. But she did not think that was the case. Yeah,
Did you run this PCR once or did you do this twice?
I only did this. I did not do the retest.
So what kind of tissue did you take
This was mouse tissue from the ear.
from the?
Yeah.
Was it possible that you actually did not get the tissue? Somewhere, like when you put the tissue into the tube, so maybe the tissue was up somewhere, it was actually not into.
I think whenever I saw the tube, the tissue was in it. It was just, yeah. There was something in there that looked like tissue. Yeah.
Yeah, for DNA degradation, I just thought it's normally, it's seldom, right?
I mean, DNA is quite stable, and even if you have a little of it,
Oh.
PCR is so sensitive that it probably would still check
Oh.
out. But I mean, it is possible.
Right,
It is
yeah.
possible, right? And, you know, I mean, other things that could happen and you probably didn't do since there are 3 in a row, but it's very easy to accidentally, someone talks to you while you're putting the polymerase in and you forget one of the components. Right? Not saying that you did that, but
Maybe.
if it were me, that's what I would think I did.
Right, yeah. Yeah, just to all of you, so then this is a research, so then anything can happen, so everything needs to, all the time need to be very careful and, yeah. All these, so many possibilities you need to think about that.
And it's a very clean and pretty gel, so you know,
Right,
you know the
very
DNA really well. So I don't think there's, you know, a problem with getting stuff stuck in the wells.
Yeah.
And did you do the DNA extraction yourself, and how was
The
that?
DNA extract. I did not hole punch the ears in the mice. I couldn't touch the mice.
But
but-
they gave you the samples and you added the liquid to break down. the-
Yeah, yeah. Oh, question.
I was gonna ask, how'd you get such good pictures of the job?
Oh, okay. So I got this from the UV citation machine, and then I took this and I put it into Photoshop, and I basically increased the contrast. The whites came out and the blacks went super black. Yep.
The primers are very specific, so no side effect for that.
Did you expect to get a heterozygous? Were you surprised?
Yeah, I
to
think
get-
those were labeled heterozygous on the tubes. I think they were supposed to be heterozygous mice.
I see the 5, 6, and 7.
Yeah. Okay.
Thank you.
Okay, so our last presentation is Samuel. So Samuel is working in my lab during this 6 or 7 weeks. So then, and show here. Okay. Oh, where? So I need to find where her. So this is her. I need to find her. Oh my, why she has so many windows there? Okay, here. Is this one? Yeah. Human Samuel.
They're all on the desktop here, if you can get
On
it
the-
back up.
Desktop there. But yeah, for that one. it's it. I think Samuel, you can just log on to your account and then get your presentation from here. Yeah. But the problem, so then, so they, yeah, she has to go to find food to feed everywhere. I'm not sure. So then, maybe I think we can just, I can just use that computer. You can log on to your account there. Right, yeah, we do not need to use this. Oh, Oh, but then for the people online, I'm not sure, so whether,
Yeah, that's right.
yeah, I think that they can still look at that, right?
Well, if this is still open, the question is, we're just having technical difficulties in terms of activating.
Right, yeah. So then, but then, oh, yeah, just can log on, but then log on, but then I cannot exit
What
there.
if I do, maybe a find, and then just write Sam. Samuel, do you know what you named your file?
Your name, your file?
Okay,
It's my name, Samuel Wilson, final presentation.
Samuel Wilson.
that's gonna be-
Samuel Wilson.
these are all in order.
Right, yeah, so we just need to find the PowerPoint.
Oh, is it on this computer? No?
That one just drag there.
Is anyone really good at computers?
Samuel, here,
Oh, yeah, that might be it. Yeah, there you go.
which one?
That's probably the today one.
Final.
Yeah.
Yeah, today,
Yeah.
yeah.
Yay.
Yay, we got it. Yeah, but then, yeah. But I cannot touch there.
Yeah, we did that before. How did we do that? How did we do that? We're not that smart.
Yeah, it cannot go there.
Ah, no, it is not.
Oh, it is
Yay.
there. It is there.
Okay, let me take some photographs of you too.
Okay,
Oh, what about-
yeah, okay. So,
Okay, now-
okay, mask off. Okay, so Samuel has been in my lab, then my lab is mainly about stem cells and for stem cells to treat cornea and glaucoma. So then during this several weeks, I just had him so shadow almost all the procedures. So then to give him an impression about what a research lab would be. And then he also did his own project. So then I'm amazed by how careful and how, yeah, just very careful and patiently to do that. At the meantime, then he was able to analyze the data with Excel and Prism to get statistical analysis. And also he was able to sit there for hours and hours to click all those things. So that's the research. Sometimes you need to be very patient to do very boring things, but then the results would be very inspiring, right? Yeah. Okay. Yeah, so let him talk about what he learned. I hope this unique experience would have some effect in your whole life. Okay.
All right, so my name is Samuel Wilson. I go to PA Leadership Charter School, and my project is characterizing glaucoma pathology by comparing the retinal nerve fiber layer thickness of a mouse model with wild-type mice. And I was a part of the stem cell biology and glaucoma lab run by Dr. Du. And my mentors were Dr. Du and Dr. Sridhar.
I want to mention, so my postdoc, yeah, Dr. Mandy, so he has helped a lot, but then yesterday he was sick and yesterday afternoon he was tested positive. So yeah, sorry about that, he could not be here.
So like Dr. Du said, this summer I shadowed a lot, and learned a lot of things. Some of them being micropipetting, cell cultures, animal genotyping, ERGs, which is electroretinography, and optical coherence tomography, which was my actual project. So starting off with background, understanding glaucoma.
In a normal eye, there's a constant production of aqueous humor, a fluid that builds up in the inner and outer chambers of the eye. And the trabecular meshwork there would allow the fluid to cycle. So it doesn't just sit, it cycles constantly. But when the trabecular meshwork gets clogged, blocked, or whatever, something happens to it, the fluid can no longer drain the way it's supposed to. Now this causes the fluid to build up, and then it starts to increase the pressure in the eye. Now this increased pressure damages the retina here and kills the RGCs, or the retinal ganglion cells, which along with their axons actually make up the optic nerve and the RNFL. Now the RNFL starts to get thinner due to the loss of the RGCs and their axons because of the pressure from glaucoma. Now, as glaucoma damages the retinal ganglion cells and the optic nerve, vision loss occurs. As you can see in this picture here, this is what someone with normal vision would see. And then this is what someone with glaucoma would see. They lose peripheral vision, and then eventually it leads to irreversible blindness. Now, the RNFL can be measured using OCTs to monitor the progress of glaucoma and predict the vision loss. Now, OCTs. This is a picture of the machine itself being set up and used to take an image of the mouse eye. This image is the rendered image of the actual retina itself. And you can see the different layers and whatnot. This image is an image of the actual entirety of the retina with the fundus there. Now this image is the 11 by 11 grid that was generated, and it's an interface that was used to actually measure the thickness of the RNFL in this rendered image here. In this image, you can see the different layers of the retina, with this one being the retinal nerve fiber layer, which is the important one for me. The rest of these, not really important, but they're important for us, but not really for my project. And so you can see the ganglion cell layer, and these are their axons that bunch up and go all the way to the optic nerve. Now, the purpose of my project was, to compare the quarterly RNFL thickness of a mouse with the myocillin mutation in age-matched old wild-type mice. So we were comparing mice that had a mutation that causes glaucoma with age-matched mice, so other old mice, to see if the mutation or the effect of the mutation. So to quantify the effect glaucoma has on the RNFL. Then the other goal was to compare the quarterly RNFL thickness of old wild-type mice and young wild-type mice to see if age itself causes damage to the RGCs. Now our methods, the first thing we did was genotype 19 mice and separated them into 3 groups: the young wild-type mice, the old wild-type mice, and the old myoc mice. Now the myoc mice were mice positive for that transgenic mutation, myocillin, which causes glaucoma both in mice and humans. And the wild-type mice, wild-type just meaning they had phenotypes that you could find in nature. Now we took OCT scans of their right and left eyes and analyzed them. And the retina was divided into four quadrants for measuring: the upper nasal, upper temple, lower nasal, and lower temple. And then there was an 11 by 11 grid generated over it that we used to measure the thickness of the RNFL. Now, next, we took that data and we measured that data using the Bioptigen software, which generated that 11 by 11 grid. And we took that data and put it into Microsoft Excel and analyzed it using Prism. Now in Excel, you can see the 4 quadrants of the retina, the same as these: upper nasal, lower nasal, upper temple, and lower temple. And we took the averages that we got from there and put them into Prism. And then we compare that data using Prism between the young wild-type mice, old wild-type mice, and the old positive mice. Now our results. Our results found that, actually first I'm gonna explain this. This is all 4 of the quadrants added together. This OD stands for right eye, or oculus dexteris, actually half the definition. And this one, OS, stands for oculus sinister, which means left eye. So you can see the results from the right eye, all 4 quadrants of all the animals added together. And here, the left eye, all 4 quadrants of all the animals added together. And you can see, comparing the young and the old, that the young mice have a thinner RNFL compared to the old mice. And comparing the myoc positive mice and old mice, You can see that the ones positive for the mutation have a much thinner RNFL than the ones without it. Yeah. Now, in conclusion, we found that the RGCs degenerate and result in the decrease in RNFL thickness in the mice positive for the mutation. And we also saw that the OCT can be used in live imaging of retina for animals with the disease, and this can be used to assess the disease progression as well as the pre and post-effective therapy. So all that means is we can use the OCTs to actually see if the treatments are working, and also to see the progression of the disease, see how far along it is, and if the treatments are slowing it or furthering along. Oh, I'm not done yet. Then next is the gradual loss in thickness and subsequent vision loss leading to permanent blindness that we saw in the mice positive for the mutation. And then we saw that this decrease in RNFL thickness was validated by a parallel experiment that was going on, where they compared the ERG, where they took ERGs of mice with and without the mutation and compared the functionality of their retinas. And finally, our results showed no significant difference between the RNFL thickness with respect to increasing age. Probably though, that's probably because of a lack of actual subjects of old wild type mice. So if we were going to repeat this or do more, try to see some more from it, we'd have to increase the number of old wild type mice we have to see any actual significant change. And then acknowledgements, I want to thank Dr. Du, Dr. Sridhar, Dr. Yuanyuan Chen, Moira, Kira, and Professor Jose Asahel, and Dr. David Boone, Stephen Jones, Solomon Lifshitz, and the Hillman Summer Research Program. Thank you.
Sure. If we go back to the data slide. So it almost looks like the wild type old mice have, slightly more, if anything, than the wild type young mice in terms of the thickness. And if you increase the N, there's quite a bit of overlap. So it seems like it's similar. Were you surprised by that result? Did you expect?
Yes, but I also kind of expected it to be that way, because this is technically, I have slides for this. This is one mouse. So what we did was we took the 4 quarters and then added them all in the all section.
I see.
So it looks like 4 different mice, but in reality, it's just one mouse. So you can't really have any, what's the word, conclusion based off of that. Yeah.
Yeah, so initially I thought they had more of the mice measured,
Sure.
rather than when they did that, Sridhar just said, yeah, some with the cloudy and something didn't get that. And also some mice then they were not, so then did not know the genotyping. So now we are repeating, actually Andrew explained that. So if with those, then we can put those back to the data, but now actually this is just the one mouse. So with the 4 quadrants.
Yes, yeah, okay, no, that's great.
So then for that one, we could not get any conclusion actually.
Now, there's clearly a difference with mutant mice. Do you think that's big enough of a difference to be useful diagnostically?
For sure, yeah. Because if I had pictures, I could show you of the difference in actual RNFL. So if they were looking at it, they could actually pull up the 2 and compare the sizes.
Okay.
But that's just the statistical average of them.
So can you go back to the B scan image? I'm curious, B scan image, so cross-section image.
The OCT image? This one?
Yeah, this one. Actually, the left part and the right part of the image is quite different. I didn't know how.
This one?
Yeah, the right part is much better than the left part.
I'm pretty sure this right here is either the optic nerve or, yes, optic nerve.
So you've got an OCT expert here. So
Yeah.
This part has much more higher speck noise, but this part seems, much more smooth. So
It could
I
be the
didn't know
way
if
to-
you did any processing on that or you
No,
just.
Sridhar did that. So the postdoc did the OCT scanning.
Okay. So it's just explored from the bioptician system.
Right.
Oh, that's interesting. See, much more smooth than.
Yeah.
Right, yeah, maybe he got some cataract, so he had problems, but then maybe this was from earlier time, and now he is getting better to do the OCT on the retina.
Okay.
Yeah. So did you actually measure this one? Like for this one, what did you click for the RNFL?
I'm not sure if I did this one.
So this should.
I can't really tell them apart.
So Samuel, what were like some of the high points of the process or the experience?
Actually, it was putting them into Prism. I don't know why, but that was actually fun.
Yeah.
Excel was boring. And then to actually measure the RNFL, it's 11 by 11 grid, 19 animals, 2 eyes each. Yeah, she knows, that was boring. I was there for days. But yeah, Prism would probably be the best one.
He also scheduled tomorrow to do the corneal OCT and to analyze the cornea. So I just wanted him to see the difference.